is Dr. Charlie Hindmarch at Queen's University, in charge of the Translational Institute for Medicine, or TIME. Dr. Hindmarch is the Scientific Operations Director for that facility and responsible for the implementation of TIME, as well as developing the strategic plan for the new institute. So I guess they're not sitting still and waiting at all. Alongside that, of course, he also offers next-gen sequencing, bioinformatics, and CyTOF facilities for Queen's researchers, and I imagine also for people in the area. And I invite Dr. Hindmarch to tell us all about it.
Thank you very much for the introduction. I'll just start my screen share. Which for some reason is, bear with me a second, I was fortunate enough to upgrade my computer yesterday, and now it's forcing me to do all the security and privacy checks.
I think we lost him, eh? Yeah, it looks like we lost him.
Dropped out altogether? Okay,
yeah.
That's serious updates. Okay.
So maybe in the lull, I'll just mention we will be starting to plan the content for our next national meeting, which we'll be having virtually in June. So we have decided to run that meeting in a virtual format. So we will be asking for potential topics both for our next community as well as for that meeting. So if anybody has ideas of things that they would like to see discussed as a community, then please feel free to put that in the chat or send us an email.
Okay.
So I think we have back.
He's back.
I am back. I will just accept the recording and try and share my screen, which this time has worked, I'm pleased to say. So I'm sorry, I hope I haven't delayed anyone.
No worries.
So hopefully you can see my presentation screen.
There we are.
Okay, that's perfect. So thank you very much for the kind introduction, and thank you to CNSP for honouring me with this award and giving me the opportunity to really showcase what we're trying to do at QCPU. So the introduction said that I'm a member of TIME, and that's right, and I'll show how TIME and QCPU interact and why that might be useful for the delegates of this committee here to know about. I wear a lot of hats at Queen's University, so I'm an adjunct assistant professor, which allows me some academic license to be able to publish papers, mostly in the genomics, transcriptomics, and molecular medicine domains. I supervise students, of course, like many of my contemporaries, within the kind of the platform management, and also have the opportunity in interacting and trying to actually pull in funds which facilitate other researchers at Queen's, but also progress my own academic career. I was really interested to see the very complex diagram that Laurence put on about the scientific platform research ecosystems, and I'm very interested to go back to that. But my first slide, coincidentally, is a distilled version of this, which really boils it down to what we're all trying to do here. We all use really fancy, nice equipment. We pour funding and good ideas into it, and we dispense bad data and discriminate it from good data in order to be able to generate manuscripts, which of course leads to a nice feedback mechanism of obtaining funding in the future. And really, I think what we're all trying to do today is establish the role of the talented scientist within being central to these kind of core facilities, because these machines don't really work very well without us. And I think we add value to the scientific community, both locally, nationally, and internationally with our efforts. So I want to talk a little bit today about the Queen's Cardiopulmonary Unit and what its remit is. And really, we're interested in primarily heart, lung, blood, and vascular. And this grant was obtained within that remit from a $10 million CFI, which was obtained by the head of medicine, Dr. Stephen Archer, who's our scientific director. And this really allowed him to obtain funds that brought this kind of infrastructure to Queen's. So QCPU, we're in the center of campus, and we have facilities that allow us to be able to perform good science, but also to recognize expert teaching. So we have areas that are dedicated towards teaching. There's 5 of us on the team. There's myself, and there's 2 PhD trained research scientists, and an expert and well-qualified molecular biologist who's also our manager and has been very good at making sure that we stay honest in our mission. Now, I said that there was 2 different institutions that really are mirroring each other. The one is QCPU, that has the research scientists and the head of medicine, but we also have something called the Translational Institute of Medicine. And the reason why I'm mentioning this now is because it's been really useful to be able to give a name to the research platforms that we have that are outside our center. So to be able to have a virtual collection of research infrastructure at Queen's that we're able to recognize and that we're able to map these resources and expertise has been very important to our mission. And I'll tell you a little bit about that moving forward. So our business model, like many of the business models I'm sure exist here, we're CFI funded infrastructure, which allowed for the initial purchase of the equipment. And this comes with some limited warranties and even more limited salaries. So we have this grave problem moving forward where we have some lovely equipment and we have warranties that are incredibly expensive. Somewhere between 10 or 20% of the initial purchase price are given to warranties per year. And also expert PhD scientists who aren't well supported either at the institutional or the national level. We generate our funds through cost recovery. So we don't have institutional support at this moment, but the cost recovery really allows us to be able to drive revenue through the research community that's already at Queen's, and ensure that we charge a price margin that allows us to be able to pay the researchers, but also potentially pay towards the warranties and ensure maybe that some of this equipment can be replaced. And we're ambivalent to our users. We are heart, lung, blood, and vasculature, but we've recently interacted with several different departments on models that are as discrete as sequencing the genome of a large prawn species, or working with other biological models that are not involved in health research. We sell users something that we call super user packages. And what this really allows us to do is to be able to say to a research group, here's 104 hours or 208 hours in a 12-month period. And it's a fixed price, and that gives you essentially access to a suite of different pieces of equipment that exist within our center. And what's really nice about this is that we can get involved at the grant writing stage and ensure that these super user models are built in as a yearly charge. And researchers and reviewers agree that these are sort of things that pass quite easily through budget committees on grant panels as being that's an institutional cost. So one thing that we found to be incredibly useful at QCPU really is to document and track our success. Now, we've only been open for 3 years, and when I first turned up, we were wearing hard hats in here, so we really have come from zero to hero. But in that time, we boast 10 of these super users, and we're serving 24 different research groups across 10 departments, 3 faculties, and even 3 universities, so that spans internationally. We have 60 QCP scientists that assisted or mentored over 60 trainees. And these include both research staff, medical students, postdoctoral fellows, graduate students, and undergraduate students. And we're also actually the home for 5 students who actually physically sit on location. We also host 3 different graduate courses. I run one on next generation sequencing, but we have 2 others that are interacted by us, the super user, the scientists, the QCP scientists. And we've tracked and measured that we've contributed to 18 publications in the last year alone. And I'll come back to how we know that in a second. And just as a quick background, I'm sure I don't need to tell the audience here what these things are, but we have genomics, transcriptomics, and molecular medicine. We also have a site-off with a Hyperion panel added, so we can do imaging mass cytometry. We have a comprehensive microscopy suite, cell culture and cytometry laboratory. We also have a Vector 4, which is a mass spec CT scanner that allows for small animal imaging. And we are recently, or rather in the process of developing a biobank for cardiovascular diseases, which is interacting with the hospital. And really what that's trying to do is to really bridge that translational gap between our local hospital, the clinicians that work there, to try and draw revenue and have samples saved in a manner that's compatible with downstream molecular techniques such as sequencing or mass cytometry. Now, I talked a little bit about being able to capture information and why that might be important. And about 3 years ago, in my role as Scientific Operations Director for Time, I was able to interact with buying an out-of-the-box networking tool from a company called Proximify. And this tool, which is broadly called UniWeb, really out of the box, it didn't have great functionality or usability that we saw. So we spent quite a few years refining it and trying to map what we have at Queen's. And very briefly, what it allows you to do is for every researcher to be able to have a profile that's publicly facing where they have tags. And these tags are as part of a controlled vocabulary that we manage that are based broadly on CIHR terms. And these tags aren't just tagged to individuals, they're tagged to publications, but also, and importantly, research platforms. And they're the same tags. So now we have a mechanism by which we can tag research platforms that fit particular different research themes, and even be able to start collecting information of the sort of people who are using or relying on these pieces of equipment in order to be able to satisfy their research needs. So this reduces redundancy around the university, so we don't need another flow cytometer. We know we've got 5 or 6 with expert users. We now increase collaboration, and we refine the mission of ensuring that we have well-managed, well-maintained infrastructure that is sustainable at Queen's University. I just want to make a note of one of the other points in our short 3-year tenure as running a research facility at Queen's like this for core needs. Really, and I updated this slide while I was listening to Laurence talking about impact measurements. It's been really, really important for us to start working towards a strategic plan. And this strategic planning process really has allowed us to identify the challenges and actually pin to those challenges specific metrics of success that we hope to obtain within a certain fixed period, say 2 to 5 years. And also marry these with annual reports. So this is our annual report for this year that details some of this, these wins that we've had, but also tells some of the individual stories about the QCP scientists, but also the kind of successes and wins that we had in terms of publications, in terms of obtaining research grant funding. And of course, we can do a lot of this by using this tagging system within Time, this UniWeb platform, because if we place QCPU as a tag and we place that against every publication, we can quickly gather information in an instant of all the publications that have been generated in the last year that have a QCPU tag on it, and we know that they've been published out of us or as a result of that. And this is proving to be critically important because I'm starting to lead conversations with senior executive management to be able to discuss the sort of challenges we face as a standalone core facility and the kind of successes we're already achieving. And we'd be lost at sea if we couldn't accurately demonstrate the value that we brought to Queen's through our endeavor. So of course, we've got a road ahead and we have our own challenges, and securing institutional sustainability for the infrastructure, the equipment warranties and upgrades is crucial for a core facility at a relatively small university like ours. We're trying to obtain hard funding for research scientists. We're all PhD trained, we're all experts, and we all have a really, really solid understanding of the research mission of the groups we work with because we embed ourselves in them. We also have in-house grant efforts from QCPU scientists like me, where I actually work with reviewers, researchers around Queen's, and I've been co-applicants on CIHR applications as well as various small level applications that bring money not only to those researchers through my support and my cover letters, but also direct money through my laboratories. And the other top tip really is to integrate research platforms under one roof. So it's not just QCPU that we're trying to capture here. We actually know that there's infrastructure around the university that is owned by private groups that has a willingness to share these as part of a larger community. And actually that might reduce some of the risks that are hampering those the owners of that infrastructure. So I'm going to thank you once again for everything that everything you've done and the very interesting talks that we've enjoyed today. And I'm very happy to answer any questions, and either now or in the future. Thank you very much.
Thank you. It was very interesting, and in a relatively short period of time there as well. So you've really come a long ways. What as you said, 0 to hero, very short, a long ways in a short period of time. So thank you again for that. So yeah, there's a few people who have some questions there. I guess we could start with Vidhu. She was wondering about the usage if a super user supersedes their cap. Are they charged on a prorated basis, left free? How do you manage that?
Actually, yeah, so it's a good question. So super users basically become priority users in our infrastructure. So if somebody has a 208-hour super user rate, they've paid at the beginning of the year for those hours, and really they are a first-class passenger. So they get priority in queues, and they get license. And of course, there's always the problem if you have multiple super users who are vying for the same attention, and then it's a first-come, first-served basis. But ultimately, we're trying to reward and encourage support of the center, because we know that gives us some financial stability and sustainability.
Very good. Actually, I'm going to hand it over to Vidhu if she'd like to speak, rather than me just reading all these off. I think that's a more natural way to do it.
So I think it's great. Like, I think you answered it. And my question was also like, I think previously in Mickey's presentation as well, like she mentioned something about membership fee, where they get sort of like a discount over the user fee. So is it sort of similar to that? Like, is it some kind of like they're purchasing a membership fee for a certain number of hours ahead of time, and then beyond that, do you have like a prorated basis of charging afterwards?
Absolutely. So that's just how it works. So there is a reduction for taking the super user rate, and then after that, users actually go on to, if they want to purchase an extra 5 hours, for example, they just go on to the standard rate for that 5 hours. I think that what happens is that we're trying to encourage rollover and trying to keep users informed about their usage of the platform to make sure that they manage their expectations properly.
Got it. And do you have any other, like, because I saw your facility, like, especially the time that compiles variety of different infrastructure technological platforms. So do you have any other, let's say, flow cytometry core or microscopy core within Queens that might be sort of like, you know, not necessarily competition, but like even a collaboration sort of model in any of the ways? Like, do you have more than what you have?
Yes. So there is another department that has a core which offers some flow cytometry. Some of these are historical, so they, I don't think it's fair to say that they're on the way out. I think they may cater to a different audience to us. We have positioned ourselves as a biomedical because the Sony SH800 has the capacity to fit within a biosafety cabinet. So we really satisfy some of the health and safety risk problems by doing that. So I guess in that way, we're kind of like, you know, we capture a user base which is maybe different from other groups. There are other flow cytometers that are owned privately by researchers and investigators, and of course we're coming into this sort of, you know, 2017, 2018. So from the perspective of where that goes long term, I'm not sure. We'd like to encourage the model of not having so much redundancy because it's wasteful, and to encourage researchers to write into their grants that they don't need this infrastructure because they have it and they'd rather contribute to that, and we share the success.
Thank you.
Pleasure.